Hi, my name is Erin Goddard, and today I'm talking about information exchange methods and what they can tell us about brain function. The exact numbers are give or take some, but this quote captures the idea that one of the main features of the human brain is its incredible interconnectivity. If we want to understand how neural activity supports perception, cognition, or behaviour, we need to understand not only what information is represented in different areas, but how information in one area shapes responses in other areas. Today I'm going to briefly introduce some measures of information exchange and how they're different from other measures of brain connectivity. I'm then going to show you results from two recent studies to give you a bit of a taster of the kinds of insights that these methods can lead to. Firstly, here's a very broad overview of different approaches to measuring connectivity. Each of these is giving us a different kind of insight. A fundamental measure of brain connectivity is anatomical, measuring which brain regions are physically connected. Another class of methods is measures of functional connectivity from fMRI. These methods are based on measuring how responses are correlated across different voxels, either when the participant is at rest or when they're doing a task. They're most commonly static measures, by which I mean that you look at the average correlations to get a single map of these relationships, but there are some studies where they also make dynamic measures, seeing how the correlations change over time. They're also commonly univariate measures, but can be multivariate. And the final category here is functional connectivity based on MEG or EEG data. These data have much finer temporal resolution, but less spatial resolution than fMRI. And like for fMRI, there are a number of different analysis methods. Although that is really a very brief introduction to these methods, the main take-home point from this overview is that each of these different measures of functional connectivity is based on response correlations, without telling us what information is exchanged between regions. It's as if we have the phone records to know when two people were talking to each other, but we don't know what they were discussing. As an alternative approach to more specifically ask what information is communicated between regions, we can use classification analyses. Classification analyses have been used for fMRI, MEG, and EEG data, and in each case the neuroimaging data is divided into trials of two or more types, for example, two different types of visual stimulus. Data from a subset of these trials are used to learn a decision rule. Then the performance of the classification rule is evaluated on held out data. If the classifier can learn to separate two types of trial using the neuroimaging data, then this is evidence that the neural responses contain information about the difference between the two stimuli. If we look at classifier performance across many different stimulus pairs, we can summarize the information across all the stimuli in our set using a dissimilarity matrix, where each cell in the matrix is defined by how different the responses to that pair of stimuli are. This idea has been used in many studies, where it's often referred to as representational similarity analysis. A strength of this approach is that the dissimilarity matrices can be compared across brain regions or across imaging modalities or even across species. If we represent the information carried by different brain regions in this way, we end up with one of these matrices for each region of interest and each time bin. With the high temporal resolution of MEG or EEG, we can then test whether the dissimilarity matrix at one time in one brain region is predictive of what happens at a later time in another brain region. For instance, if we measure responses to a visual stimulus, we can look for times where the dissimilarity matrix in occipital cortex is predictive of later activity in frontal regions, and for evidence of feedback from frontal to occipital cortex. But how will we know if this is really an exchange of information or just similar independent processes in two regions that evolve over time? A really useful framework here is that of Granger-causality, where we make sure that any predictive power is greater than what we can predict from the past within the destination area. There's still the possibility that there's a third untested area that's linked to both regions, but I think that Granger causality gives us a great starting place for trying to measure information exchange between regions. There are many potential ways to implement this. A couple of examples from the literature are to use partial correlations, which is what I've used in previous work, but there's another really nice approach of a hierarchical GLM, which you can read about in this PNAS paper from Tim Keitzman and colleagues. Before I move to some experimental results, I'll just try to give you an intuition for how these methods work, showing you the example of the method that I use. First, we start with the dissimilarity matrices in our regions of interest at a particular time. Then we go back to an earlier time and ask what was happening in those regions of interest previously. To calculate the feed-forward flow of information from the occipital to the frontal region, we take the correlation between these two matrices, shown in blue, partialing out any correlation with the frontal region at the same earlier time. And this gives us a single measure of the feed-forward information flow. To estimate the feedback information flow in the reverse direction, we correlate these two matrices, again partialing out the correlation with the occipital region at an earlier time to give us another single point. We then repeat this entire process over time until we end up with time courses of feed-forward and feedback information flows. So in the remainder of my time, I'm going to show this method applied to two projects. In this first project, we were testing the effects of spatial and feature-selective attention. We created novel object stimuli varying in colour and shape. On each trial, participants saw two of these objects, and their task was to fixate on the central cross but covertly attend to one object and to report either its colour or shape. We included all attention conditions across different blocks with many trials of each. We collected MEG data on 20 participants, defined two regions of interest, and performed a series of classification analyses over time. Using the same trial data, we trained classifiers to discriminate 4 separate stimulus dimensions. The classifier analyses gave us data like this. Here you can see the average decoding of stimulus colour using data from the occipital region of interest across 4 different attention conditions. During the initial part of the stimulus response, classifier performance is high, regardless of attention condition, but over time these traces diverge, so that only in the attended location, attended colour condition is there sustained information about stimulus colour. We also have the same data for the frontal region of interest, where there's no early peak, but we do see decoding of the task-relevant information at a later time. And now we can repeat the same analyses for the decoding of object shape. And here you can see there's a broadly similar pattern of decoding across our two regions of interest. I'm now going to move to the results from the information flow analysis, but before we move on, I just need to break this data down a little further. If we take just a single attention condition, what I've been showing you so far is the average classifier performance, averaged across all classification pairs, but we have the data for each step size separately, and we use this multi-dimensional quality in the information flow analysis. Just to reiterate, we're using the classifier performance as a measure of the stimulus-related information that's represented by these areas, and we're testing for evidence of Granger-causal relationships between these over time. But we have more than the 4 time courses shown here. Instead, we have 24 time courses in each case. So here I'm showing the difference between the feed-forward and feedback information flows over time. To make these easier to interpret, here I've plotted the same difference data as a colour map on top of the plot of average classifier performance in occipital cortex. You can see that in this initial period that I've highlighted with the box, information flow is dominated by feed forward, but then there's a period that's dominated by feedback. I'm now going to show you the same results for decoding of stimulus shape. Now, although these figures look a bit different to those for colour, there are some similar features. There's an initial period of feed forward, followed by a period of feedback. If we look at these together, you can see there's timing differences quite clearly. But note that in both cases, the onset of feedback information flow corresponds with the time where classifier performance for different attention conditions diverges. This provides extra support for the idea that frontal cortex is driving the attentional selection of task-relevant information. I'm now going to move to this final example. This study involved a visuospatial working memory task. There were 8 possible items that were presented at 4 of 9 possible locations. And on each trial, participants saw 4 items presented sequentially, followed by a maintenance period and then a retrieval item. Their task was to say whether the retrieval item was one of the 4 in the encoding set. We had 4 regions of interest, including some visual regions, prefrontal cortex, and posteromedial cortex, which was our main region of interest for its potential role in visuospatial working memory. And we used classification analyses to decode each stimulus location and each stimulus identity. Here you can see the classifier accuracy for decoding location. The red lines indicate stimulus onset, so here we're looking at decoding the location of the first encoding item. We calibrated the task difficulty for each participant, so the shaded regions are where the data is averaged across fewer than the total of 11 participants. And we have this decoding data for each of the encoding stimuli, and also for the retrieval stimulus. We also have similar time course data for decoding of stimulus identity, which was lower overall, but in the interest of time, I'm going to move straight on to the information flow analyses. Here you can see the information flow between PMC, our main region of interest, and occipital cortex, across the different task phases. The dark grey shows a null distribution of values where we ran the analysis on data with shuffled trial labels. As before, we looked at the difference between these 2 directions of information flow. So you can see more clearly that occipital to PMC dominates when the visual stimuli are presented, but there's some evidence of PMC to occipital information flow dominating during this latter part of retrieval. As an extra test of whether these information flows are meaningful, we also looked at the correlation across subjects between information flow and task performance. In this case, none of these correlations were significant. But I'm now going to show you the same results for a different pair of regions. This time we're looking at information flow between PMC and prefrontal cortex. Overall, the information flow from PMC to prefrontal was higher than the reverse, and we also saw some evidence that the degree of this dominance was correlated with better task performance across individuals. But it's important to note that these correlations didn't survive a correction for multiple comparisons, and we do have a relatively small sample size of just 11 participants. So we see these correlations with behaviour as not definitive, but potentially as a motivation for future research. And more generally, I think this approach of correlating information flow with behaviour could be a fruitful way to test the behavioural significance of information flows. In summary, I think that measures of information exchange give us the potential for more targeted and specific insights into brain connectivity. In the 2 examples, I've shown how timing of information flows can be related to other features of the data, and correlations with behaviour provide an interesting future direction for these methods. I'd like to finish by thanking my co-authors, the funding body, and the Symposium organisers for putting this together. And lastly, if you're interested in these approaches and these kinds of research questions, we'd love you to apply for this postdoc position.